The way that blood circulates around a nephron is important. And if you look at this, all of the kind of the, I don't know, yellowish or gold color tube that you see in this slide is the nephron itself. And then you can see the blood vessels around it. Now, there's the beginning part of the nephron is called the glomerular capsule, and that's this part right here. And you notice inside that little capsule, there is a tiny little capillary bed called the glomerulus. And then, so blood goes through these little capillaries in here, and then the blood leaves and comes out and goes through another capillary bed that surrounds the rest of the nephron. So this is technically another little portal system where blood goes through one set of capillaries and then through another. Now it's a tiny one. It's not like the one that's sending blood from your GI tract to your liver. Those are significant capillary beds on either side. This is a really tiny one, but it still is technically a portal system. And so you can see the blood flow. You've got capillaries surrounding the nephron, and that blood flow is really important because material is both leaving the blood and entering the nephron, but the nephron is also returning some of that material into the blood, because basically at the first step we're going to take too much good stuff out of the blood, and so a lot of that has to be recovered and put back in, and we have to keep the bad stuff out but put some of the molecules that we need back into the blood. There are 6 steps to forming urine in the nephron, and each of those steps occurs in a particular location within the nephron. So we're going to walk through the whole thing from the beginning to the end and see what happens at each step as the urine is being formed. Step 1 takes place in the renal corpuscle, and the glomerulus is inside there, and so those two things are interconnected. This is where the ultrafiltrate is formed, and it's formed passively. It's just diffusion that causes it to happen. So the ultrafiltrate is not urine, but it's sort of pre-urine. It's urine before all the final adjustments have been made. It's very similar to blood plasma, but you don't have blood cells. You don't have most proteins or other large molecules because they just can't really get out. So the material from the ultrafiltrate has left the blood. It's now in the nephron. And what's in the ultrafiltrate is actually some important stuff that we want to hang on to. Water, there's a lot of mineral ions, there's glucose, fatty acids, amino acids, some vitamins, and then the waste products that we actually want to get rid of, and primarily urea, because that's the one that we're really talking about here. So all the stuff that's going to wind up in the urine is in the ultrafiltrate, but what we're going to do with the rest of the nephron is recover the things that we want to hang on to, while keeping the toxic things still in the urine, keeping that out of the blood. So the first part that we run into after we pass the renal corpuscle is the proximal convoluted tubule. And here you've got both active and passive reabsorption happening. So some of it is just diffusion, but some of it is active transport. So this has a huge amount of surface area in it, again, and down at the microscopic level, just to speed up the transport of materials across that surface. So ions, mineral ions, organic nutrients, and the small proteins that are in the ultrafiltrate are moved out of the ultrafiltrate using active transport. So that's burning ATP. Water is leaving the ultrafiltrate by osmosis. And again, that's because, you remember, osmosis is a form of diffusion. That's happening because the salt concentration is fairly high outside the nephron. So that means the water concentration is higher inside the nephron than outside the nephron. So water is able to diffuse out of the proximal convoluted tubule. When we get to the end of the proximal convoluted tubule, we have already recovered all of the organic nutrients that we are going to recover and all of the small proteins that we're going to recover. Um, about 60% of the sodium and chloride ions has been taken out of the ultrafiltrate, and we've recovered it, and about 60% of the water. So you notice that we've already done a big percentage of the job just when we get, by the time we reach the second step here, we've already done an awful lot of it. The loop that goes down and then comes back up, that part of the loop of the nephron, used to be called the loop of Henle. They're always trying to get rid of structures that are named after people, and Henle was a person. So they just call it the loop of the nephron, although it's not really catching on. But anyway, if you see loop of Henle or loop of the nephron, it's the same thing. So when we're in the descending limb of the loop of Henle, more water is leaving the ultrafiltrate by osmosis, and that's because the salt concentration is high outside the nephron, so that means the water concentration is high inside the nephron, so the water is diffusing out. By the time we get to the bottom of the descending limb of the loop of Henle, about 25% of the water is remaining in the ultrafiltrate, so we've recovered a lot of the water. The ascending limb of the loop of Henle looks pretty similar, but it functions very, very differently. The walls of the tubule are waterproof, so there is no osmosis through that tubule. What you do have is active transport of sodium out of the tubule, and that is basically what's making the salt concentration so high on the outside of the tubule, is that it's being actively transported out of the ultrafiltrate, and so it's high outside the tubule. By the time we get to the end of the ascending limb of the loop of Henle, we've reabsorbed 75% of the water that was originally in the ultrafiltrate. 90 to 95% of the sodium and chloride ions that were in there have been recovered, and 100% of the nutrients have been recovered. Step 5 takes place in the distal convoluted tubule, and in the distal convoluted tubule, more water is leaving the ultrafiltrate by osmosis, so that's passive transport. By the time we get to the end of it, we've got anywhere from 5 to 20% of the original water remaining in the ultrafiltrate. And then there's more active transport of sodium and chloride ions out of the tubule, so we're still, the little bit that's left is still being they're still being actively transported out of the tubule and back into that interstitial fluid that's around the tubule. Step 6 it takes place in the collecting tubule, and if you look at that collecting tubule in the picture, you notice there's a bunch of little branches where tubes have been cut off, and that's because many distal convoluted tubules drain into a single collecting tubule. So you've got a bunch of nephrons that are emptying into one collecting tubule. And what the collecting tubule does is make the final adjustments to the concentration and volume of the urine. And it does this by controlling the permeability of the wall of the collecting tubule. And that needs a little bit of explanation. If the permeability is high, that means that water can pass through the tubule wall more easily. And we're still in the medulla where the salt concentration is high, so the water concentration in the medulla is lower than it is in the collecting tubule. So as long as we kind of open the doors and make it so that tubule is permeable to water, water is going to leave the tubule and you're going to recover it. And that, of course, makes the urine very concentrated when you do that. Less water and whatever's in the urine is more concentrated than it would otherwise be. If the permeability of the tubule wall is low, then little water is lost in the collecting tubule, and lost means lost from the ultrafiltrate. And so that means the urine has a higher volume, and so, of course, it's more dilute. What controls the permeability of the collecting tubule wall is a hormone called ADH, which stands for antidiuretic hormone. If the ADH levels circulating in the blood are high, then the tubule wall is permeable, more water is reabsorbed, and it makes the urine more concentrated. ADH is released from the pituitary, and it's basically part of the mechanism that controls your blood volume. So if your blood volume drops, that's the signal that says, okay, we better start hanging on to water. And so ADH is released, and you basically produce more concentrated urine. Now, alcohol tends to dehydrate you, and that's because of what alcohol does to the release of antidiuretic hormone. If you drink alcohol, alcohol inhibits the release of ADH, which means that you're going to produce a lot of urine, and it's going to be very dilute, and you're going to tend to dry out because of that. Well, this is basically what we just said. So here's and you're measuring your blood volume. Well, it's actually in the hypothalamus, which sends a signal to the anterior pituitary. So depending on your blood volume, you either release ADH from the anterior pituitary or don't release it, and that will help to adjust your blood volume either up or down depending on what it's doing. This is a nice summary slide that just goes over everything we just talked about. So this is actually a good one to look at for review. I won't go through it again, but it's what we basically just talked about at each step.